Hey there, micro friends. Welcome to another episode of Let's Talk Micro. As always, I am so happy that you're here with me. And if this is the first time you're listening to Let's Talk Micro, well, thank you for choosing it as one of your podcasts. Okay, so let me talk about the episode real quick and then we'll go ahead and jump into it. Well, as you know, one of the challenges of antimicrobial resistance, or AMR, is drug discovery. And we have a lack of therapeutic options, and you don't see many new classes of antibiotics being discovered. And what you see coming out there is additions to existing classes, and you see things like beta-lactam, beta-lactamase inhibitor combinations. But what if AI could help us? So in this episode, I am joined by Dr. James Collins from MIT. And in the episode, he talks about the work that his team did using AI and ultimately ended with some promising candidates that show activity against Staph aureus and Neisseria gonorrhoeae. So let's go ahead and listen to the episode. Hey listeners, so as you know, one of the challenges with antimicrobial resistance of AMR, right? It's the discovery of antibiotics, and there's not enough of them. And in this podcast, I've done episodes where you see like potential antimicrobials. So I have shown different types of research, and this one caught my eye as I read the article because it involved AI, or artificial intelligence. So with me today, I have a guest to discuss, some compounds that were discovered using a deep learning program. With me, I have Dr. James Collins. Dr. Collins, welcome to Let's Talk Micro.
Thanks for having me.
My pleasure. So I think before we dive into the episode, maybe we can start with a little bit of an introduction. You can tell the audience about what you do and anything else that you want to share with them.
Yeah, so I'm a bioengineering professor at MIT. I also run a lab at the Broad Institute of MIT and Harvard, as well as the Beast Institute at Harvard. And our lab has two dominant areas of interest. The first is around using AI to both discover and design new antibiotics. And this builds on our now 2 decades worth of work using machine learning in the context of network biology to better understand how antibiotics act and how resistance arises, really with the goal initially of boosting our existing antibiotic arsenal. And then the second area of interest for our lab is synthetic biology. So using engineering principles primarily to re-engineer microbes, endowing them with synthetic gene circuits and other molecular components, enabling those microbes to have new functions to address a range of applications.
Well, thank you for being here again. And one thing that we see, like I work in clinical microbiology, and we see how AI is being, it's making its way into the lab and it's been applied to instruments. As thing is, especially with things like microscopy, we see it in parasitology and things like that. So I ask you, why did you decide to do AI for antibiotic discovery instead of relying on more traditional methods?
Yeah, we got intrigued on this possibility now going back about 6 or 7 years ago. MIT was launching a major institute wide new initiative around AI and had a symposium to announce this effort. And I had a chance to sit next to Regina Barzilay, one of our faculty stars in AI, who had been at the time working on a number of deep neural networks in the context of chemical synthesis and compound analysis. And we realized we both had interest in drug discovery and thought about could we use deep neural networks specifically in the context of antibiotic drug discovery, and formed a collaboration and saw we had an opportunity to use these tools really as an efficient means, an efficient tool to explore much larger chemical spaces than we had the means to do experimentally. So as you allude, the traditional means would be to collect curate a library of chemical compounds that you would then screen one by one against a bacterial pathogen of interest to see which one exhibited antibacterial activity, taking the most promising molecules for additional studies of mechanism of action, target confirmation, resistance, etc. We instead said, let's create a small library of compounds that we could use as training compounds. We initially then screened these against E. coli, identifying those that were antibacterial, those that were not. Used the data about whether they were antibacterial or not, along with their structural information, to then train a deep neural net, which we now could point to enormous chemical libraries, including those consisting of over 1 billion compounds, which we could never have screened empirically in our lab, but we were able to screen a fraction of these, a substantial fraction, in this case, 110 million molecules in just 3 days, going back a few years ago. So AI, in this case, were leading us to models that were just tools, but important tools, that significantly increased our efficiency and allowed us to explore a much bigger part of discovery space.
Yeah, that is well said, and that's one thing that we see when AI is being applied to like things as I mentioned with instruments and methodology, that it's just it's helping out dramatically, like taking out, especially when it comes to screening negatives, like you train it to screen negative samples, and then it limits that, and then that way your technologists can focus on other tasks and use a better use of their time. So let's go ahead and get a little bit technical, and maybe for those of us in the clinical microbiology side, as I read the article, I just want to go over some of the terms that I saw, and maybe we can talk more about them. So your team used something called graph neural networks as part of the pipeline. Maybe in simple terms, can you explain how your graph neural networks or GNNs help in predicting antibacterial activity?
Yeah, so in both our earlier work going back 5 years ago, and then in our most latest piece that we published in Cell, we used graph neural nets, a form of a deep neural net model, in order to train a computer to look at a compound structure and associate it with a desired feature. So in this case, these models are actually able to look at a chemical structure from a graphical standpoint. So it can consider bond by bond, so say connecting two atoms, collections of the bonds, and associate it with, in our case, being antibacterial or not antibacterial, or being toxic or not toxic. You can then feed it some number of these compounds. We initially trained it on 2,500 compounds. We now have been training it on 40,000 compounds or so, enabling it to look at all of these substructures and begin to now have the ability to look at a completely new structure that it had never seen before, and based on its graphical structure, make a prediction of would it make for a good antibiotic or would it make for a non-toxic drug compound. We did this 5 years ago in the context of antibiotic discovery, applying that model to large libraries to try to discover new compounds that could be antibacterial in nature. And in this new piece, we actually applied that model initially to look at fragment libraries. So fragments effectively being substructures or rationales of compounds. In our first effort using generative AI, we looked to use these fragments as our starting point. And so to identify amongst many millions of possible fragments, which ones could be good starting points, we actually applied our trained graph neural network to these that could then score them on a 0 to 1 scale, 1 being very highly likely to be an antibacterial fragment, 0 being highly unlikely, to identify those around which we could begin our generative efforts where we could decorate these fragments to try to make new antibiotics.
Okay, and then also there's something called chemically reasonable mutations. So I maybe can talk a little bit about how this approach, expand fragment into new molecules and why is it so powerful.
Yeah, so this is an AI model that's based on genetic algorithms. So it's an older model. We didn't invent it, but we utilized it in the context of our study, called CREM. And CREM standing, as you alluded, chemically reasonable mutations. And what you can do is start with a chemical structure. It might be a fragment, it could be a whole compound. And as the name implies, you then begin to introduce mutations to that structure, those that are reasonable from a chemical standpoint, so that it still looks like a molecule that might make for a good drug, or it could be a molecule that you could synthesize. And then at each stage that we're doing that, we're also evaluating against our AI model calculation of, did those chemically reasonable mutations lead to a compound that was predicted to make for a good antibiotic? And now, was it predicted to be a better antibiotic than what you had started with? And we did this repeatedly in the context of this CREM as applied to both our fragments as starting points, as well as to the second part of our study where we actually didn't use the fragments as starting point, but started with atoms, say an oxygen, nitrogen, and carbon. Okay, from there, what's your reasonable mutation? You're going to have to begin to build out structures and then begin to apply CREM.
Okay, and before I ask you about the compounds and things like that, there's one more term that I would like to have the listeners learn more about, which is your variational autoencoders, and how has this helped in exploring chemical space and generating novel antibiotic candidates?
Yeah, so this was the second AI generative model that we used. This was first effort on CREM was done by Jackie Valeri. Our entire study was led by Artie Krishnan, postdoc in our lab. Jackie did much of the modeling work around CREM, and then our variational encoder work was done by Wengang Jin, who's a young professor, Northeastern, former postdoc, here at MIT and a great colleague of ours. And with variational encoders, what you can do is start with a atom or a compound structure, and you begin to decorate that with additional different bonds, molecule substructures. And again, at each stage, evaluate it against our trained model to see, did those changes that you introduced lead to an interesting new molecule that was predicted to make for a good antibiotic or was predicted not to be toxic? And from there, we, in each of these, got to many millions of interesting looking possibilities.
And for the listeners, I'm going to put on the show notes, I'm going to put the publication so you can read it with more detail as we talk about this. So then, as I was reading, right, so I saw that there were like 24 synthesized compounds, and out of those, like 7 show activity. Maybe you can walk us a little bit of how you narrow those to those most promising candidates.
Yeah, it was interesting. So we, again, started with these 2 different generative approaches, arrived really at millions of possibilities. In fact, many, many millions, but we did a number of filters. So the most important was identifying those that are predicted to make for good antibiotics to have antibacterial activity as evidenced by our models. The next was predicted to be not toxic. So you don't want to be toxic to human cells. You want to wipe out the bacterial pathogen, but spare the human cells. The third was then additional drug-like properties, so-called Payne's scores and Brink's scores. So to ensure that they're not highly promiscuous, to ensure they don't have other features that may not be very good for drug-like properties. And then finally, we looked at synthesizability. You need to be able to make the molecules. And that's probably where the biggest challenge was. I think the models that were led by our colleague Connor Coley here at MIT, I think were really quite good. But it turns out that it's really quite challenging to synthesize these molecules. From those filters, we arrived at many hundreds that looked exciting. And Artie Krishnan, our lead postdoc, reached out to probably about 10 different chemical synthesis firms. And they basically choked on the list we gave them. They more or less said, we can't do this. And I think it was probably too overwhelming in the extent. And then already narrowed that down to maybe of order 100 or so. And Etamine, a chemical synthesis company based in the Ukraine, Kiev, actually said, hey, we can do this. And came back and said we could make 40 of these. And in the end made 24 of them, which was, I thought, a pretty good success rate. And as you alluded, of those 24, 7 actually turned out to exhibit significant antibacterial activity, which is really quite a high hit rate of order about 30% or so.
Okay, and before I ask you about the 2 lead compounds, because as I read the article, right, so one of them showed activity against Staphylococcus aureus, and then the other one showed activity about Neisseria gonorrhoeae. Just as a microbiologist, I'm curious, and I know that both of them, they're significant, and they're both, there's resistance when it comes to Neisseria, narrow choices. We see all these organisms. Staph aureus has its own challenges. So, do you select those 2? You went out, or you found the compounds and then you tested organisms and found they had activity against those specific ones?
No, we had picked those organisms at the outset. So we, in fact, collected training data against Staph aureus, and we collected training data against gonorrhea, really with the goal of designing molecules that be effective against MRSA, methicillin-resistant Staph aureus, as well as molecules that would be effective against drug-resistant gonorrhea. And there's needs for both, because MRSA is probably the most famous antibiotic-resistant pathogen, and unfortunately, multi-drug resistant, even PAM-resistant gonorrhea is back and present in our communities, including here in Massachusetts, where I'm speaking from. And we advanced those efforts and were able to show that we could design molecules with those pathogens in mind, and to have molecules that actually were quite effective against those pathogens.
Okay, yeah, I think I do remember reading about, was it from Massachusetts, that they found like one Neisseria gonorrhoeae that was like very resistant. I
It's
think it was
PAM resistant, so it's basically
yeah.
resistant to all the antibiotics that we use in treatment, and it's very concerning, of course.
Yeah.
So we need these early discovery efforts to then also translate into effective development efforts.
So after you went through all that process, as I mentioned, NG1 and DN1 were the lead compounds. So as far as as microbiologists, we like to know, right, how they work, mechanism of action. And this podcast, we have talked about the current antibiotics, and some target DNA, some target RNA synthesis, some target protein synthesis. So what are the mechanism of action as far as NG1 and DN1?
Yeah, we were able to work out on each of these. So, they acted both at the membrane level, and then on NG1, we were actually able to get after a protein target and work out in some detail. So we are still working on DN1 as where its protein target might be, but in each case, they had membrane activity in slightly different ways, but really quite exciting that we could get after the targets quite reasonably with some of the approaches we were getting after.
Okay. And this, this work in mouse models, and that's just, that's significant. So can you talk about more about, what that means for their potential as future antibiotics?
Yeah, it's interesting. I probably wouldn't characterize these per se as leads, but as early hits, promising hits from our effort. And we were able to show that they worked in vivo models, both against gonorrhea in a vaginal infection model and against MRSA in a skin infection model. Again, promising, right, that you're getting after at least some activity. I think in each case, there's a lot more work to be done for kind of nailing down optimizing their properties so that they could be considered to be drugs. And that's where I think we need two things. I think we do need to advance AI models to help us with optimization, with analog generation. And I think we need better and more extensive investment in the field. There's, I think, growing engagement from the microbiology community, growing engagement from the AI community towards early discovery efforts, in part by efforts around AI, deep learning, new tools to bring to bear. But we need to be able to take these early hits through preclinical development and actually advance them into trials. And that's where I think we need a lot more work.
Yeah, and that alone is a very lengthy process from the time that something shows the right that activity and then to that ends up being an actual drug on the market. That's a very lengthy process, many
It's
years.
lengthy and it's expensive.
Yes, definitely. Well, this approach, it's right kind of opens like a new uncharted chemical space. And we have talked about how AI, it helps us, how we can screen this, massive amount of compounds that you wouldn't be able to do normally, or would take a long time, and it does it in a short amount of time. So, anything else can you add about this approach and its meaning for the future of antibiotic discovery?
Yeah, I think what's exciting is multiple fold. I do think it's bringing a whole new generation of young people into the space. I think it's bringing computer scientists and bioengineers into the antibiotic space. So we need more talent. I think we're seeing it come in. I think we're seeing the world community get engaged as this is a meaningful application of AI for good. I do think AI is allowing us to explore much larger chemical spaces, which of course will get us after new chemical matter, which will have new mechanisms of action, which can overcome existing resistance. It won't overcome resistance in all time, but existing resistance. Thus, I think giving us a leg up in the fight, the battle of our wits against the genes of superbugs. So I do think that AI as a tool is helping usher in a new golden age of antibiotics. And if we can support that with the needed investment to advance the most promising molecules, I think we can do quite well against the emergence of these resistant pathogens.
So as we kind of start coming to a close, I mean, anything else? I mean, you by now you have mentioned it pretty much, but just to kind of just wrap it up and put it in one place, as we say, how do you see AI reshaping the way we discover and develop antibiotics in the fight against AMR? Anything else you can add?
I would say that I do think it's early in the use of AI in drug discovery, including antibiotic drug discovery. I think the groups that are going to make the real breakthroughs in the coming decade will be the groups that do three things. One is collect their own data, both to train the models and validate the models. Two is the groups that have recruited and trained a multilingual team. And by multilingual, I mean a group that can have young folks that can speak to microbiologists across to artificial intelligence experts to medicinal chemists to bioengineers, so that you have young folks that speak more than one disciplinary language. And then third is, I think, the groups that properly integrate human intelligence with machine intelligence. We still need the human in the loop in order to deal with these complexities. And I think AI is really still not just, but is an important growing in power thought partner. But it's the groups that can ask those right questions and use AI smartly that I think are going to make the real the real breakthroughs in AI drug discovery in this next decade.
Excellent. And I like how you mentioned, sometimes, we hear AI, and maybe sometimes someone might have the thought of, right, this whole thing about we see in movies and we go right there, our minds go to there, are taking over or not, but we do need the humans. And so far, some of the applications that is being it is helpful. And some of us, we use some version of it in our daily lives. And it's not to tell us what to do, but it kind of helps us on reshaping a problem, or we have a question, how to, an email or things like that. So it's definitely very exciting to see what's coming down the line. And
Yeah,
as you said, there are still some challenges left, and it's still early on, but it's exciting.
yeah,
Uh,
yeah. I agree.
and I like to ask this question to our guests. We all tend to have that microorganism that we like for some reason. Either has some significance because when we were in school, we did a paper about it, or it was the subject of our research, or something visual, a mechanism. Do you have a favorite microorganism?
I would probably say it's E. coli. I trained as a physicist and did my work initially in medical engineering, so I was not trained in microbiology, wasn't trained in molecular biology. But our initial efforts during the late 90s in synthetic biology, and then our initial efforts in systems biology, were in E. coli. So an organism that very well studied, genetically tractable, relatively easy to manipulate, but for which we still have much to discover and much to be harnessed. So I'm a big fan of E. coli.
Thank you. I like it. I think that's like you're the first person that has mentioned E. coli. Yeah. Well, Dr. Collins, thank you so much for being here, and I appreciate it. As I said, I saw this on social media, and it caught my eye, and I'm like, oh, I'm gonna give it a read, and then reach out to you. I thought it would be a good topic for our listeners. So, before we wrap the episode, is there anything else that you want to tell the audience or about this?
Not so much of this. I think I assume you have a lot of young folks on the audience. I would just say I'd say that despite all the challenges we're facing right now in American scientific community, that I think it's a great time to be a young person. I think there's so much to be discovered, so many exciting projects to go after, particularly both with the new technologies that are emerging that are impacting microbiology and the challenges that we face. And so we need young talent to be motivated and excited. And I'm excited for them, and I hope they are as hopeful as I am for them, in spite of all the challenges we face for different reasons.
Yeah, well said. And that's something that I mentioned. Even for me on the clinical side, it has changed so much in the past like 10 years, and we continue to see more and more changes. So, once again, this has been great, and I love how you explain everything and how the audience likes it. It's like easy to understand. So, thank you so much for taking the time to be in Let's Talk Micro.
Well, thank you, Liz. Thanks for having me. Enjoyed talking with you.
All right, my pleasure. Well, there you have it, micro friends. I hope you enjoyed this episode, and this is such an interesting topic. And as Dr. Collins said, AI is still in the early stages for this application for like antibiotics, but still a fascinating topic. Okay, so as always, you can find Let's Talk Micro on all podcast platforms like Apple, Spotify, Amazon Music, GoodPods. So I invite you to subscribe, download episodes, and if the app allows you to do so, please go ahead and leave a review. As for our social media, you can find us on Instagram, TikTok, YouTube, and Blue Sky Social as Let's Talk Micro, on X as Let's Talk Micro 1, or LinkedIn as Luis Plaza. There's the website, letstalkmicro.com, and also an email address, which is letstalkmicro@outlook.com. I definitely invite you to check out the website. It has some great resources, episode updates, and more. If you want to support the podcast in any way, you can do so via a Venmo link or via Buy Me a Coffee KOFI, both links available on the show notes. And if you are looking to learn more about antibiotics, please check out www.learnantibiotics.com and the Learn Antibiotics book available on Amazon. These resources include cheat sheets, practice tests, games, and more, and they are being used by thousands of people worldwide and may be helpful for you or your colleagues. Great resources from Dr. Timothy Gauthier, a very passionate pharmacist. And the Let's Talk Micro podcast is supported by the American Society for Microbiology. To learn more about microbes, visit asm.org. And that, my dear audience, it's the end of this episode. I hope you enjoy learning about AI and these compounds NG1 and DN1. As always, I enjoy sharing this information with you. Such a fascinating topic, and if AI could be applied to this, maybe this can help with the fight against AMR. So, stay tuned. Great things coming your way. As always, I am so grateful for the support. We are about to hit a download milestone. So this is a great time for you to go back, see if you missed some episodes, download those. And hey, maybe you forgot some, or maybe you want to go back to some content. So download them. As always, continue bringing that passion to what you do. It's so important. You do such great work. So, as always, stay motivated, stay safe, and of course, continue talking micro. Until the next time. Bye.